Under conditions of antigen or antibody access, small, soluble complexes tend to predominate. But with roughly equivalent amounts of antigen antibody, precipitates are always formed that are going to lead towards the lattice formation. Such precipitates activate the inflammatory response and probably underline some types of occasional energies, for example, farmer's lungs. Complexes between antibodies and large antigens, for example, nucleic acids, are associated with systemic autoimmune diseases, such as systemic lupus erythematosus. In the presence of complement, only small complexes are formed. In fact, C3 can solubilize larger complexes, and SLE is commonly associated with C2 and C4 deficiency. So let's discuss the characteristics of immune complexes. We have blocking, C1q complement binding, inflammation, lysis, phagocytosis, FcR complex formation, cytotoxicity, and B-cell memory production. The blocking of T-cell or antibody-mediated killing by complexes in antigen or antibody axis may account for some of the unresponsiveness of tumors or parasitic infections. Well, in C1q complement binding, the first component of complement binds to the Fc portion of complexed antibody, possibly under the influence of conformational changes in the shape of Ig molecule. Although some workers hold that occupation of most sites that are combining with IgG molecule is all that is needed. Activation of classic complement pathways takes place after that. The inflammation is simply breakdown of products of C3 and C5 through interactions with mast cells, polymorphs, etc., and they are responsible for the vascular damage that is a feature of immune complex diseases. Well, lysis occurs in bacteria that requires the complete complement sequence. Sometimes C567 units moves away from the original site of antibody bindings that are going to activate C8 and C9 complexes and cause the lysis of innocent cells, for example, red blood cells. So this whole procedure is known as reactive lysis. Talking about phagocytosis, it is mostly done by macrophages, polymorphs, and eosinophils. The characteristics of this phagocytosis is Fc and antigen binding that is going to complement and make them strongly opsonic. The other example is the IgM binding. For example, it binds to complement much more than to the cells. And the last one is RBC binding, in which the most complexes in the circulation are packed up by the red blood cells via their complement receptors. All these three are occurring thanks to the phagocytosis. Well, Fc receptor complex formation occurs when there are several types of receptors that bind Fc constant part of antibodies. Some are found on phagocytes and facilitate uptake of IgG opsonized bacteria. Others are present on mast cells and bind IgE. The interaction of IgE and specific antigen then triggers mast cell degranulations. A different Fc receptor on B cells bind antigen-antibody complexes and act to switch off further antibody production. Let's discuss about the cytotoxicity. Well, when antibody bound to a cell or microorganism contacts Fc receptors, the result may be killing rather than phagocytosis. Cells able to do this include macrophages, monocytes, neutrophils, eosinophils, and natural killer cells. And the last one is B-cell memory production, in which complement receptors on the follicular dendritic cells help them to retain immune complexes and present the antigen to B cells in a way that by selecting for mutants with high binding affinity encourages the increase or maturation of the affinity of the antibody response. So, we just talked about the characteristics of immune complexes. Stay with us for a brief summary.